in one of the desks? You want to sit down here? Come on down. Raise your hands first. Whoa. Hi, how you doing? You all hear me? All right. So this is going to be the seating arrangement. So remember your seats, please. Makes it easier to take attendance. It would kind of suck if you got marked absent because you sat in the wrong seat. Plus any group work. So now that I have a seating chart, I can make up groups so we can do some group work, some problem solving stuff. I want you to do me a favor and check your email settings for Moodle. Some of you have been emailing me about things that I've posted on Moodle. So I posted things about the Panoptos and about the seating chart, and you were sending me questions via email indicating to me that you had not gotten the message. So check your email settings and make sure it's set up for you to get group messages so that when I post something on Moodle, you'll get it. Yes? Moodle emails not working at all for any class? No. All right. So I'll have to use regular email. Or Ravens. Would you like to get a Raven? I'm just kidding. So the Panopto should be, yes, it's recording right now. So it's set up to automatically record, so I don't have to remember. The one from Wednesday did not show up on Moodle. It somehow got lost. Nobody can find it. So I re-recorded a Camtasia and put that on Moodle. So basically I did the lecture from home with my 2 dogs and my bottle of wine. So that is available. The Panopto from Monday did record. Some people have said that there's problems with the audio. I played it and it played fine for me. So I don't know if it's your computer or what. Any questions on anything? If you're unhappy with the seating chart, I don't know what I can do unless you can find somebody to switch with you. If you're in a seat that you're definitely not happy with, find a classmate who's willing to switch. I don't know what else I can do. Any complaints about the room that you're in? How many of you there are? How many of me there are? Forward them to the president. I have no control over that. The only thing I can control is what goes on in the classroom. Questions? Anybody? Before we start. All right. So we talked about nerves. Now we're going to talk about muscle, the other electrically excitable tissue we're going to deal with. Muscle works by the same principles. The only thing is now we're going to add in a contraction apparatus. Muscles contract, nerves don't, but we still have the same resting membrane potential, action potential, depolarization to threshold, etc., that we've been dealing with so far. Depolarization to threshold. Threshold here is opening of voltage-gated calcium channels, which then stimulates release of calcium from the sarcoplasmic reticulum. Skeletal muscle fibers have large, well-developed sarcoplasmic reticulum, so they can store a lot of calcium. 90% of the calcium for contraction for skeletal muscle fibers comes from what's stored in the sarcoplasmic reticulum. This little guy here, phospholambane, we're not going to worry about him for right now. When you want muscle cells to relax, there's two ways of getting calcium out of the cytoplasm. When calcium enters the cytoplasm, there's contraction. When you remove calcium, there's relaxation. You can pump it back in by primary active transport into the sarcoplasmic reticulum using a calcium ATPase. You can get it out of the cell by secondary active transport. Yes?
Can you slow down a little bit, please?
Sure.
Thank you.
I'm going to go through this all again.
Okay.
Don't worry. So this NCX sodium calcium exchanger, right, secondary active transport, as sodium comes in, diffusing down its concentration gradient, calcium is removed from the cytoplasm. So those are the two mechanisms to get rid of calcium, either primary or secondary active transport. Of course, the secondary active transport occurs because the sodium gradient has been established by the sodium-potassium ATPase. And we're going to see that over and over and over again in pretty much every organ system we talk about. We're not going to talk about the mitochondria. There is some calcium storage in mitochondria, but it's used for other things. It's not necessarily important for contraction. So the function of skeletal muscle first is, of course, to allow you to move, provide support, and allows you to move around. It also happens to be the biggest producer of heat in the body. Heat comes from metabolic activity, and skeletal muscle is highly metabolically active, right? It has to use lots of ATP for contraction, generates a lot of heat. So the majority of the heat in your body is generated by skeletal muscle. Anybody want to guess where the second most comes from? Where? What's something that's highly metabolically active even when you're sitting down? Circulatory system. The heart beats a lot, uses a lot of ATP, generates a lot of heat, but it is not number two. Good guess. The brain. Yes, the brain is functioning constantly. Uses a lot of glucose, generates a lot of ATP, generates a lot of heat. And skeletal muscles also have nutrient storage. If you're ever under a period of starvation, your muscles will start to waste away because your body will be taking all the proteins, breaking them down into amino acids, so you can convert the amino acids to glucose for energy. So it's a storage form for amino acids. I hate this thing. Anatomy. Now this I'm going to fly through because you should already kind of have some idea about this. Muscles are made up, the cells are long individual fibers. They can be up to 3 inches long, 3.5 inches long. So these are cells that are long, stretched out, so they are multinucleate. They have multiple nuclei, not just one. And that's because the nucleus is there for protein synthesis. Muscles need lots of proteins constantly. Contraction is based upon actin and myosin. So having multiple nuclei in these long cells allows you to have protein synthesis, not just in the center of cells and then try to transport it this long distance, but everywhere along the length of the cell. So that's why the cells are multinucleate. They contract because of this organization of the contractile unit of skeletal muscle, the sarcomere. Sarcomere is the distance between two Z-lines. Z-lines are where these thin actin filaments attach. So contraction occurs because myosin, we talked about myosin being a motor protein in neurons. Myosin, when it's activated and breaking down ATP, the heads move. So they grab the actin filaments, pull them towards the center, which we call the M line, the middle, and that generates contraction. So a sarcomere, or a whole muscle, can contract to about 60% of its resting length. And that's about what it shows here. So proteins that we have to deal with are going to be the thick filaments made up of myosin with all these little heads here. And then the thin filaments made up of actin here. And then actin is kept separated from myosin. Myosin can only bind to actin when the site, the binding site on actin that myosin binds to, is available, is open. There's a protein called tropomyosin, which covers up that active site. And so calcium causing muscle contraction, its function is to make sure that tropomyosin moves so that actin and myosin can come together and cause a contraction. Talked about ion concentrations for various cells. We've been focusing on nerve cells up until now. Ion concentrations for skeletal muscle cells are pretty close to what we've talked about for neurons. Right? Extracellular fluid, 145. Intracellular fluid, 12. Potassium, the same. Hydrogen ion we haven't really talked about yet. We deal with that in pH. It does contribute a little bit to resting membrane potential, but not enough for us to worry about. Chloride, we said chloride was variable for different cells. For skeletal muscle, chloride is actually higher in the extracellular fluid than it is in blood plasma. Bicarbonate. The bicarbonate in the interstitial, the bicarbonate concentration in interstitial fluid of muscle is a little bit higher than what you have in blood plasma. In blood plasma, it's between 22 and 26 millimoles. It's about 27, 28. That's because when skeletal muscles contract, their waste products are acid. So the bicarbonate is there to buffer the acid to make sure that there's no pH change. We talked about this A- part. We said that for electrically excitable cells like neurons, the reason that they have a resting membrane potential that's negative because potassium is leaking out constantly, and because there's a large concentration of negatively charged anions, which are mostly proteins and amino acids and organic acids, inside the cell that you will not find in the extracellular fluid. Any questions? Yes? Negatively charged organic anions. So things like amino acids, proteins, other organic acids. Yes? That's the thing I hate about this diagram, is they don't give you a range. They just give you the center of the range. But it's pretty close to what we talked about for blood plasma. Just a couple of, so the chloride's a little higher, and the bicarbonate's a little higher. But other than that, the sodium and potassium have the same ranges. Yes? What is the acid? Speak up.
What's the acid that's produced from contraction of muscle?
There's a bunch of different ones. The one that is most common is lactic acid. So the general rule in physiology is always that the electrical event causes the mechanical event to happen. So you have to have an action potential before you can release a neurotransmitter. You have to have an action potential before a skeletal muscle contracts. So the electrical event happens before the mechanical event. So if we look here at the action potential, right, there's a short delay between the action potential and contraction. That's because there's a number of steps in between that. The action potential has to cause calcium levels in the cytoplasm to rise such that tropomyosin can be moved out of the way, actin and myosin can bind, and start going through what's called a cross-bridge cycle. Tension generally develops slowly because you have to activate all of those myosin heads on those thick filaments. You have to get the tropomyosin out of the way so that those myosin heads can bind to actin. That takes a while. So it takes a while to generate tension. So you'll see here is the contraction phase. Then when calcium is removed from the cytoplasm, it starts to relax. Now the stimulus for all of this is neural. So the alpha motor neurons in the spinal cord, we talked about large alpha motor neurons. They send signals down to skeletal muscle fibers. The action potential causes release of acetylcholine. Acetylcholine is the neurotransmitter associated with muscle contraction. Acetylcholine binds to ligand-gated ion channels on the skeletal muscle fiber, which allows sodium to enter. Now those ligand-gated channels, those acetylcholine receptors, are nicotinic. Nicotine is an agonist for those receptors. The other acetylcholine receptors that we'll talk about are associated with the autonomic nervous system. So they're muscarinic receptors, and their agonist is muscarine. So two different types of acetylcholine receptors. In this case, acetylcholine will bind to the nicotinic acetylcholine receptor on the skeletal muscle, causes the channels to open. Sodium travels through the channels from the extracellular fluid into the intracellular fluid, into the cell. That's depolarization. So the cell depolarizes, depolarizes to threshold, generates an action potential, voltage-gated calcium channels open, and then we get contraction. And we'll go through all those steps in detail later. One of the things I want to get into is what is called quantal release theory. Basically, you have all these storage vesicles inside of neurons, and they all contain neurotransmitter. According to this theory, every single one of those vesicles contains the same concentration or the same amount of neurotransmitter. So we said every action potential is the same, right? You can't make bigger or smaller action potentials. So according to quantal release theory, every action potential causes release of the same amount of neurotransmitter. The same number of vesicles are released from the cytoskeleton every time. So action potentials are all the same size, and the amount of neurotransmitter that's released for every action potential is the same. So the only way to increase stimulation of this postsynaptic cell, this skeletal muscle fiber, is to increase the number of action potentials per millisecond or per second. So you have to increase the frequency. You can't change the amplitude. You can't change the size of the action potential. So in order to release more acetylcholine, you have to have more action potentials. Any questions on that? Does that make sense? So this, where a motor neuron stimulates a skeletal muscle fiber to contract, is referred to as excitation-contraction coupling. You may see it abbreviated sometimes as ECC, EC. Essentially, the steps are, we talked about acetylcholine release from the neuron. Acetylcholine binds to nicotinic acetylcholine receptors. Sodium enters the cell. Cell depolarizes to threshold. Action potential is generated by opening of voltage-gated sodium channels. Now, the histology of skeletal muscle cells, they have specialized portions of the cell membrane called T-tubules. The T-tubule is basically the cell membrane invaginating or puckering into the cell. So the T-tubule, this T part here, right, travels down into the cell. So you've basically got the membrane of the cell invaginating or going into the cell so that you can get those voltage-gated calcium channels close to the sarcoplasmic reticulum. Because the action potential, remember in the neuron, the action potential, which was sodium, traveled down the axon. When it got to the axon terminus, it stimulated voltage-gated calcium channels to open, and they caused what to happen? Yeah, neurotransmitter release. So we've got the same thing. We've got an action potential caused by sodium stimulating voltage-gated calcium channels, but we're not getting neurotransmitter release in this case. We're getting calcium, voltage-gated calcium channel stimulating release of calcium from the sarcoplasmic reticulum. So calcium from the sarcoplasmic reticulum, you will get some from the extracellular fluid, but the vast majority is what's stored inside the cell. They will bind to troponin. Troponin's an interesting protein found in skeletal muscle and in cardiac muscle. One of the tests you can do to see if somebody had a heart attack is to test for the presence of troponin in the blood. Now that would give you a false positive if you had recently suffered any muscle damage. If you tore a muscle or you were working out pretty intense, you might get some skeletal muscle troponin that spills over into the plasma. So we have this wonderful way of distinguishing between the troponin that you find in skeletal muscle and the troponin you find in cardiac muscle. So we can tell if it's a muscle injury, a skeletal muscle injury, or if it's an actual heart attack, if there's been heart damage, based upon the type of troponin that you find in the blood. Calcium binding to troponin. Troponin is a calcium binding protein, and calcium binds to it. Troponin makes tropomyosin get out of the way. Remember we said tropomyosin was this protein that's covering up the binding sites on actin. So when tropomyosin is no longer covering the binding sites of actin, myosin and actin can get together and do their thing. And their thing is contraction. So that's going to trigger what we call a cross-bridge cycle. And we'll go through that. It's a cycle of myosin binding to actin, pulling on actin so that those Z lines come closer together, contraction, letting go of actin, grabbing onto another, pulling it closer, this cycle of grabbing actin and pulling. Removal of calcium by active transport, either primary or secondary. Primary if you're getting it back into the sarcoplasmic reticulum. Both primary and secondary if you're just pumping it out to the extracellular fluid. Removal of calcium causes calcium to be released by troponin. Troponin is no longer pulling on tropomyosin, so tropomyosin returns to its regular conformation where it is blocking actin and myosin interaction. And the muscles relax. Capiche? Questions? Is this bringing back memories? Kind of? Yes?
Action potential is operating ACH fine. This show I know. Sodium potassium. Going from the sarco level down to there's like still an influx of sodium into the muscle cell.
Yeah, so up here there are, at the synaptic cleft here, this is called the active zone, where the neuron and the postsynaptic cell, the target cell, are close together. So the opening of nicotinic acetylcholine receptors causes depolarization because sodium can come in. But then right next to or right outside of the synaptic cleft are voltage-gated sodium channels. So that's where the action potential comes from. And of course that travels in a wave and opens more voltage-gated sodium channels. And it travels down to the T-tubule to open voltage-gated calcium channels. Did that answer your question? Okay. Now all cells have sodium, potassium, ATPase, so this is going to be active in all cells. So the cross-bridge cycle, the actual act of getting the Z lines to be pulled closer together or towards the middle of the sarcomere. When you start out, you've got troponin blocking actin and myosin binding. When troponin moves out of the way and myosin can bind to actin, myosin in the resting state is going to have ADP bound to the head. ADP is adenosine diphosphate. Yes?
Instead of this blocking these,
Yeah, calcium binds to troponin, moves tropomyosin out of the way. Yep. Did I say that wrong? Okay. If anything's ever unclear, let me know. These big lips are hard to move sometimes, so me and Mick Jagger just got this problem. So in a resting state, myosin has ADP bound. When it binds to actin, it's going to eject the ADP. Get out of here. Right? As it does so, there's going to be a conformational change. So there's going to be a hole where the ADP was. The myosin head is going to contract. It's going to pull the actin fiber. So that if the Z line is out here, this movement from about 120 degrees here to about 65 degrees there, that movement from obtuse to acute, math terms, right? Causes, so that if the Z line is out here, then this is going to be pulled by the myosin moving. If ATP is present, when would ATP not be present? When you're dead. So normally, throughout your entire lifetime, your muscles should be able to do this. Because you should always have ATP inside your muscle cells. The only time you don't have ATP is when you're dead, and then the myosin can't let go of the actin, and you end up with something called rigor mortis. But since you're alive, the ATP binds to the myosin head, right? So that hole that ADP was occupying now gets occupied by ATP. And that basically shifts the myosin head back to that 120-degree conformation. So it causes it to cock. That's called recocking or cocking. That's accomplished because ATP is hydrolyzed or broken down into ADP and organic phosphate. So splitting that third phosphate bond provides the energy to cock the head back to 120 degrees, and then you get the ADP, or you get the PI that gets released first, and then you're back up to here. And then the ADP gets released, and there's a conformational change. That's called the power stroke. ATP comes to fill in the hole, gets hydrolyzed to ADP and PI. That energy allows the head to cock. And then the PI is ejected, you go back, and you just keep going through these 4 steps as long as there's calcium in the cytoplasm, until you get maximal contraction. Once those actin thin filaments start bumping up against one another, the sarcomere can't get any shorter. Questions? So let's talk about pharmacology, since this is a pharmacy school. There is a number of ways to interfere with what normally happens between neurons and skeletal muscle fibers at the neuromuscular junction. A lot of toxins from venomous creatures, scorpions, snakes, spiders, acts at the neuromuscular junction, which is why a lot of the features that you'll see from people who've been bitten by a very venomous creature are going to be paralysis or involuntary muscle contraction, right? One or the other. Paralysis comes in 2 varieties, flaccid simply means that you can't move anything, your muscles are relaxed. Spastic means that your muscles are always contracting. You still can't move because your muscles are contracted and not relaxing. Now, most of these toxins are going to cause a flaccid paralysis, so they're going to block transmission between the motor neuron and the skeletal muscle fiber. But there are some toxins that will stimulate that and cause a spastic paralysis. So, blockers of neuronal sodium channels. How would that cause a flaccid paralysis? How would that prevent you from contracting the muscles? Yeah, no neurotransmitter release. Up here, the action potential has to stimulate voltage-gated calcium channels so that exocytosis can occur. If they're blocked, voltage-gated calcium channels never reach threshold, you can't release acetylcholine. Right? So tetrodotoxin and saxitotoxin do this. Tetrodotoxin is the one we talk about all the time, TTX. It's in every physiology book you've ever seen. That's our prime example of a sodium channel blocker. And it comes from fugu. We know what fugu is. Pufferfish. Yes. So all you sushi fans, stay away from the fugu. Unless you know the guy that's preparing it for you has done it thousands of times successfully before. Otherwise, it could be nasty. Conotoxin blocks voltage-gated calcium channels, again preventing neurotransmitter release. Without calcium, you can't have exocytosis. Conotoxin comes from scorpions, I believe, as does dendrotoxin, which acts on potassium channels, and that basically prevents you from repolarizing. Or excuse me, it keeps the channels open so that you can't generate an action potential. Yes?
So we know the name of the toxins.
No. I just want you to know the mechanisms. I want you to understand what the channels are doing. The ones that you should know are tetanus and botulinum. Most of you probably know this already. If you don't, you're going to get it in micro, and you may talk about it in PDA. Tetanus toxin stimulates acetylcholine release. It directly stimulates exocytosis. So tetanus toxin causes tetanus, which is spastic paralysis. The muscles contract and cannot relax. Botulinum toxin does the opposite. It directly blocks neurotransmitter release and prevents release of acetylcholine so that skeletal muscle fibers will be relaxed and not contracting. So botulinum toxin is injected in order to relax facial muscles so that wrinkles disappear. Right? Botox. Imagine what would happen if you mixed them up. That'd be a bummer. Walk around like this. All your facial muscles. Acetylcholine esterase. Acetylcholine esterase in a cholinergic synapse deactivates acetylcholine. It breaks down acetylcholine into acetate and choline. If you block acetylcholine esterase activity, acetylcholine stays active in the synapse longer, causes an increase in skeletal muscle contraction strength. The only one you might talk about in PDA would be physostigmine, maybe neostigmine. Those are really, I think, the only drugs that are prescribed. They're usually given to people with multiple sclerosis, so that it increases the contraction strength, increases their ability to move. Other things that will block this, if you're fans of Homeland and other shows like that, neurotoxins from chemical weapons generally block acetylcholinesterase. Acetylcholine receptor channel blockers and activators. All right, so the agonists we talked about are acetylcholine and nicotine. The antagonists, curare, which is, and bungarotoxin. Curare, I think, comes from frogs, poisonous frogs. Some of the tribes in South America put it on their darts for hunting. Both of those block opening of acetylcholine channels, so you get a flaccid paralysis. And then blockage of sodium channels on muscle cells so that you can't generate an action potential. Tetrodotoxin, which blocks sodium channels on neurons as well. Saxitotoxin, which blocks sodium channels on neurons as well. And conotoxin, which is another type of scorpion venom, I think. You had a question? No? All right. Really, the only ones you need to remember are tetanus and botulinum as far as the toxins are concerned. But you have to understand the mechanism. So what's going to happen if you block sodium channels? What's going to happen if you block calcium channels, etc. Energy sources for muscle contraction. Obviously, ATP is the energy source. You make ATP via glycolysis and the Krebs cycle, the electron transport chain, so we call that aerobic. Right? Because you use oxygen for that. You can get energy out of phosphate bonds, so the energy in one phosphate bond is about 7.3 kilocalories, which is 7.3 food calories. ATP gets stored in muscle via something called creatine. So creatine phosphate stores phosphate. So what happens then is as you use ATP, it can be replenished by simply transferring phosphate from creatine phosphate to ADP, making ATP. That's referred to as substrate level phosphorylation, because you're simply transferring phosphate from one molecule to another. If you're a sprinter, your creatine phosphate system will be better developed than in other types of athletes because you've trained in order to be able to make more creatine phosphate so that you can maintain your high level of activity for a short burst. Right? Because this stuff lasts for about 10 seconds, 10, 12, 15 seconds max. Once you've used your creatine phosphate system, the next thing to do is to use the anaerobic pathway. The anaerobic pathway will allow you to continue at a high level for a couple of minutes. In the anaerobic pathway, glucose is converted into lactic acid in the process generating ATP. This all happens in the cytoplasm. This is the glycolysis portion of anaerobic respiration. The generation of lactic acid, the buildup of potassium, the loss of ATP will all contribute to fatigue. What you would like, if you're exercising, unless you're training to be a sprinter or a power lifter, you would like to stay away from this anaerobic pathway and get into this aerobic pathway. In order for that to occur, you have to make sure that oxygen is getting to the cells fast enough for them to continue aerobic respiration. So the only thing that limits the aerobic pathway is the availability of oxygen and the availability of glucose. (9,999 times out of 10,000, it's going to be oxygen that is lacking. It's almost never glucose if your liver is functioning normally. So in the aerobic pathway, you take glucose and you can make vast quantities of ATP because you've got oxygen available for the electron transport chain in the mitochondria. So vastly superior to anaerobic respiration. Muscles prefer, their preferred source of energy is fat. Under normal resting conditions, as you sit here right now, you are burning fat. Your muscles are getting about 2/3 of their energy from free fatty acids in the blood. They're taking those fatty acids out of the blood and they're breaking them down into pyruvate, which can enter the Krebs cycle. That's because you get more energy from fats than you get from glucose. Right? You get twice as much energy from a fat molecule as you would get from a molecule of glucose. So skeletal muscles prefer to use fatty acids. As your level of exercise intensity increases, they start to use less fatty acids and more glucose because it takes longer to break down fatty acids than it does to break down glucose. So as exercise intensity increases, the amount of fat you're actually burning during exercise decreases. You're actually starting to use more and more blood glucose. But that should not interfere with your exercise plans, because the good thing about it is once you're done exercising, your muscles shift back to fatty acid, and what they have to do now is they have to replenish all those storage, the storage of glucose as glycogen and everything. So after exercise is when you burn the most fat. About an hour to 2 hours post-exercise is when you get your actual fat burn, not actually during exercise. Different types of muscle fibers. It's based upon their metabolism, how quickly they fatigue. Muscles that don't fatigue very easily are called slow oxidative fibers, and they're red. Anybody ever wondered what's the difference between dark meat and light meat? Right, on chicken? Well, dark meat is these slow oxidative fibers. And they're red because they contain myoglobin. Myoglobin is an oxygen-binding protein that's related to hemoglobin. So these cells are able to store oxygen inside their cytoplasm bound to myoglobin. They have a slow myosin ATPase activity, so that means that their cross-bridge cycle is slower than in fast-twitch muscles, which means that they don't burn as much energy as quickly. Right? There's a slower cross-bridge cycle. And these fibers are generally smaller. When you get to the fast glycolytic, now the term glycolytic simply, so oxidative tells you aerobic respiration. Glycolytic tells you anaerobic respiration. So these fast twitch fibers, right, so these slow oxidative are slow twitch fibers. These fast twitch fibers are white. They don't contain myoglobin. They're there to generate fast, powerful contractions. So the myosin ATPase is fast, cross-bridge cycling is fast, uses energy fast, burns ATP very quickly. The fibers are large. Fast twitch fibers are designed to generate maximal contraction strength in a short period of time, whereas slow twitch fibers are designed to generate mild contraction strength over long periods of time. So muscles that are muscles that are mostly slow twitch would be things like the postural muscles, the muscles that hold you up. The soleus in the calf underneath the gastrocnemius is the muscle we talk about mostly. It's about 80% slow twitch fibers. Fast twitch fibers are things that you need to generate fast or rapid contractions. Your eye muscles are almost 100% fast twitch fibers. Because if you're watching a tennis match or whatever, you have to be able to watch that ball, right? So those are fast twitch muscle fibers. The bad part about that is they are easily fatigable because they focus primarily on the creatine phosphate system for energy and anaerobic glycolysis. They can generate powerful contractions, but only for a short period of time. In the middle, we have these guys. Fast oxidative glycolytic. Ooh. They can go either way. If you decide that you want to run marathons, well, these fibers will start to act more like slow oxidative fibers. They'll start to accumulate more myoglobin. And they'll act more like slow twitch fibers. If you decide that sprinting or powerlifting is going to be your thing, well, these fibers will then start to act more like fast twitch fibers. They'll start to accumulate enzymes for using creatine phosphate and anaerobic glycolysis for energy. Questions on skeletal muscle? So the next best thing, or the next closest thing, I should say, to skeletal muscle is cardiac muscle. It's striated like skeletal muscle, so it has arrangement of actin and myosin inside the cells the same as skeletal muscle. The cells are smaller, though, so they only have one nucleus. They don't extend over long distances. They don't have to because they're connected to other cardiac muscle cells by gap junctions. So essentially, any depolarization that occurs in one is going to travel to all the cells that it's connected to. There is no neuromuscular junction. The synapse that is going to stimulate contraction of cardiac myocytes, cardiac muscle cells, is electrical. It's an electrical synapse. There's no neurotransmitter released, so there's no neuromuscular junction. And it is involuntary, meaning that you don't have to think about it. It'd be kind of a bummer if you had to think 70 times per minute. Contract, relax, contract, relax, contract. That would suck. You'd never get anything done. Cardiac muscle action potentials are similar, but different. This part here in the beginning looks a lot like a normal neuronal action potential, and then it gets weird. Cardiac muscle action potentials are longer in duration than neuronal action potentials or skeletal muscle action potentials. That's because they have a specific type of channel called an L-type calcium channel. L stands for long. So the calcium channel stays open for a long period of time. And so that's what this plateau here is. You've got this fast sodium here, like any other action potential we've talked about so far. Starts to repolarize, but the calcium coming in negates the effect of the potassium leaving. So you get this kind of flat plateau where calcium is coming in, potassium is leaving, and there's no real change in membrane potential. Then when the calcium channels close, the fact that potassium is leaving repolarizes the cell. So this prolongs the absolute refractory period. What did we say the absolute refractory period was for? What happens during the absolute refractory period? Nothing, right? During the absolute refractory period, you cannot generate an action potential no matter how big the stimulus is that's stimulating the cell. Just nothing happens. That's because the sodium channels that are going to be generating this rapid depolarization don't reset until you get back down to resting membrane potential, or close to it. So the absolute refractory period is extended by this calcium plateau. And that prevents tetanus. Right? Skeletal muscles can go into tetanus. If you have high-frequency stimulation, acetylcholine is always being released, the muscle cell will constantly contract. Let me show you what heart tetanus looks like. Not good. In order to fill up with blood to pump for the next beat, the cell or the heart has to relax. So the absolute refractory period prevents tetanus in the heart. So it makes sure that your heart continues to contract and relax normally. Then, of course, there's going to be a relative refractory period because of a brief hyperpolarization, which is not shown in this diagram. So essentially, we divide this up into phases. So phase 0 is the rapid depolarization caused by opening of voltage-gated sodium channels. So that's going to give you what we call a spike. Right? That's the pointy part of the action potential. It looks like a neuronal action potential. Phase 1, after the voltage-gated sodium channels close, you start to get a brief repolarization. Then it flattens out because voltage-gated potassium channels and voltage-gated calcium channels are both open. So any potassium leaving is balanced out by the calcium coming in. So you get this plateau to extend the absolute refractory period. Calcium channels close at the end of phase 2. So phase 3 is then the repolarization phase, where voltage-gated potassium channels are still open. Potassium's leaving the cell. It's returning to resting membrane potential. Phase 4, the voltage-gated potassium channels close. The cell is back close to resting membrane potential, and you've got normal potassium leakage, maintaining resting membrane potential. Now, normal resting membrane potential for cardiac muscle cells is negative 90. So it's farther away from threshold. But when we look at the electrical conduction system, you'll see why that's not a problem. Yes? Yeah, so phase 2, the calcium channels are open. When they close, phase 3 is repolarization, because only the potassium channels are open now. Yes? No, that actually marks the point between phase 2 and phase 3. When voltage-gated calcium channels close, phase 2 ends, phase 3 begins. So excitation, contraction, coupling in cardiac muscle is different than skeletal muscle because there's no neuromuscular junction. It's via gap junctions. It's an electrical synapse. So now we can go back to this. So look at this beginning portion in phase 0. What is different about that compared to what we talked about for a neuron? What don't you see? Yes. Threshold. Right? You don't see a slow depolarization to threshold opening of voltage-gated sodium channels. The sodium channels open like that because the cell immediately reaches threshold. The reason it immediately reaches threshold because it's connected by a gap junction. It's an electrical synapse. So there's no slow depolarization to threshold. You've got an action potential traveling from one cell to the next through gap junctions, that electrical synapse. Again, the action potential travels through a T-tubule found in both skeletal and cardiac muscle. Again, opens voltage-gated calcium channels, which stimulates opening of calcium channels in the sarcoplasmic reticulum. Now, cardiac muscle gets more calcium from extracellular fluid than skeletal muscle, but it still gets about the majority of it comes from sarcoplasmic reticulum. Of course, to get it back in, we're going to have the same mechanism. So really, this is pretty much the same as the skeletal muscle mechanism. Calcium binds to troponin, contraction. Calcium is removed by a calcium ATPase pumped into sarcoplasmic reticulum or pumped out of the cell. And then the NCX, the sodium calcium exchanger, can pump calcium out of the cell. You get relaxation. Relaxation, then actin and myosin can no longer bind. So it's essentially the same as what we talked about with skeletal muscle, except for one thing. It's electrical synapse instead of a chemical synapse. Now, smooth muscle is different. That's the black sheep of the family. Somebody's always got to be different. Not striated. Not striated because the actin and myosin filaments are not all lined up on the same plane. Right? For cardiac muscle cells and skeletal muscle cells, when they contract, the cells get shorter. When they get shorter, they will actually bulge out in the middle because you're taking all of those components and squishing them into a shorter cell. So the cell, as it contracts, will actually become a little bit thicker, a little bit wider. With smooth muscle cells, you've got these actin filaments connected to dense bodies in the plasma membrane, so proteins that are embedded in the plasma membrane, rather than Z-lines. So the cell can not only get shorter, it can get thinner. Contraction can happen on all planes, all angles inside the cell. Right? So when the smooth muscle cell contracts, it gets smaller in all dimensions, not just shorter, like skeletal and cardiac. And that's what makes it a smooth muscle cell rather than a striated muscle cell, because when you look at it in the microscope, you can't see the orderly arrangement, the overlap of actin and myosin filaments, because they're all arranged in different directions. They have within them caves or caveoli, little caves, which are basically puckering of the membrane. And that puckering allows for a number of things. It could allow for a synapse or any number of other things to occur. Smooth muscle can come in two forms, single unit or multi-unit. Single unit is the most prevalent. That's what you're going to find in the GI tract, blood vessels. Single unit smooth muscle, the muscles are connected together by gap junctions. So when contraction is stimulated, they contract as a single unit. The entire tissue contracts. So those types, that single unit smooth muscle is found in the lining of the GI tract so that you can keep stuff moving along by peristalsis. It's found in the lining of blood vessels so that when blood vessels need to dilate or constrict, the tissue contracts as one. Multi-unit smooth muscle is found in very few places in the body. The best example is probably the eye. The muscles controlling the diameter of the lens. Those are all multi-unit. So in multi-unit smooth muscle, each individual smooth muscle cell contracts on its own. There are no gap junctions connecting the cells, so they can't contract as an entire single unit, as an entire tissue. They each individually contract on their own. Excitation, contraction, coupling in smooth muscle is more detailed. It's a little more complicated than what we talked about with skeletal and cardiac. There are a number of stimuli that can stimulate the cell. It could be a neurotransmitter. It could be an endocrine substance, a hormone. It could be stretch. They have a lot of mechanically gated calcium channels. It could be any number of things. But the common element is entry of calcium into the cell. Calcium entry, there are no T-tubules in smooth muscle. So calcium entry is going to cause diffusion of calcium through the cytoplasm, which causes opening of calcium channels in the sarcoplasmic reticulum. So the majority of calcium for contraction of smooth muscle comes from the extracellular fluid. There's not a lot of sarcoplasmic reticulum in the smooth muscle cell. Calcium activates an enzyme called calmodulin. When we talked about calcium, we said there are a number of functions. It stimulates exocytosis. It also activates certain enzymes. One of the enzymes that activates is calmodulin. When calmodulin is active, the calcium binds to calmodulin, activates it. It can activate an enzyme called myosin light chain kinase. Unlike skeletal and cardiac muscle, the myosin that you find in smooth muscle has to be phosphorylated in order to be active. Myosin in cardiac and skeletal muscle is always active. The only limiting factor for contraction there is the ability for myosin and actin to bind. In smooth muscle, the myosin has to be activated by phosphorylation. So calmodulin activates an enzyme called myosin light chain kinase, or MLCK for short. Now the state of the smooth muscle cell, how much of it is contracted or whether it's relaxed, is determined by the balance of two enzymes. Myosin light chain kinase, which is phosphorylating myosin and activating it, and myosin phosphatase, which is removing phosphate from myosin and deactivating it. So whichever enzyme's activity predominates is going to be the state of the smooth muscle cell. As myosin light chain kinase is activated, its activity is going to become greater than myosin phosphatase, and the cell is going to contract. If myosin phosphatase activity is greater, the cell is going to relax. But again, contraction and relaxation is stimulated by either getting calcium into the cytoplasm or pumping it out of the cytoplasm. Once myosin is activated, it can start going through a cross-bridge cycle. And the cross-bridge cycle is pretty much the same. The only difference is, instead of having one phosphate binding site, there are two. Right? So there's the phosphate binding site for activating myosin, and then there's the binding site for ADP and PI. But once myosin is phosphorylated, the cross-bridge cycle takes the same, right? Myosin binds to actin, get ejection of ADP and PI for the power stroke, right? Actin gets pulled on by myosin. ATP binds to myosin, causing release. Hydrolyzed back to ADP and PI, causing it to cock or go back to normal, and then cycle through. As long as there's calcium in the cytoplasm. Questions on that? Yes? Yeah, dephosphorylation of myosin. So when calcium is removed from the cytoplasm, myosin light chain kinase is deactivated. Right, myosin phosphatase dephosphorylates myosin, and then there's relaxation. Now, smooth muscle can maintain a contraction and enter what's called a latch bridge cycle, which is not really a cycle. It's once the latch attaches, it stays loosely bound to the actin, so it can maintain a contraction over a long period of time. That is not found in skeletal or cardiac muscle. Yes?
You said there's two active sites on myosin.
There's the activation site where it's phosphorylated, and then there's the normal binding site where ATP binds. Yeah. Yes?
Could you repeat the latch cycle? What did you say about latch?
Oh, in the latch bridge cycle, essentially what happens is myosin and actin stay loosely bound to each other, so you can maintain a contraction for a long period of time. It doesn't go through a complete cross-bridge cycle. It enters what's called a latch state. And that allows, like arteries, when they constrict, it allows them to maintain that constriction for a long period of time without having to use a lot of energy. So it's a unique feature of smooth muscle. It's especially important in the vasculature. Smooth muscle has a unstable resting membrane potential, and it's closer to threshold. We said that cardiac muscle is at minus 90, which is far away from threshold, but it doesn't matter because it's an electrical synapse, so it automatically reaches threshold anyway. Right? Action potential simply jump from cell to cell. Smooth muscle, the resting membrane potential is much closer to threshold. It's about minus 55, minus 60. So you can get what are called slow waves. So as you get this unstable resting membrane potential, you get threshold is reached. There are some action potentials produced as long as threshold is exceeded. Then when it dips back down and hyperpolarizes, you don't have action potentials. Right, so it goes through this cycle of slow depolarization, which could be subthreshold. If it reaches threshold, then there are some action potentials produced as long as the resting membrane potential is above threshold. And then slow hyperpolarization or slow repolarization. All right, so you go back to the midpoint of resting membrane potential, and then you hyperpolarize and go farther away. So this unstable resting membrane potential allows for there to be spontaneous contractions in smooth muscle. It's especially important in the GI tract to keep things moving. Right, so smooth muscle in the GI tract, you will get periodic waves of contraction to make sure that stuff is being pushed from where it came in to where it's supposed to go out, so that nothing gets stuck. That's a bummer. So this is kind of a summary of the differences between the three types of muscle. As we go through various organ systems, we will call back to this stuff, and we will talk about the function of cardiac muscle when we talk about the heart, the function of smooth muscle when we talk about the GI tract and the vasculature and other things. Essentially, skeletal muscle, as we talked about, is striated, right? Has a neuromuscular junction. Cardiac muscle, striated, no neuromuscular junction, it has an electrical synapse. Smooth muscle, not striated, multiple ways of stimulating it. Questions? Bless you. Okay. What I'm going to do is I'm going to record a camtasia for synaptic and junctional transmission. A lot of the stuff that I talked about for skeletal muscle and for smooth muscle will be revisited in this as well. So I'll post a camtasia for this, and I'll let you go. Right now, we spent a half an hour getting the seating arranged. So, see you Wednesday. I will be posting the tutoring information, schedule for tutoring and review sessions on Moodle, probably this afternoon. So if you want to check. And from now on, I will send you messages via normal email rather than Moodle until we figure out what's going on. Hey. Yep. Yeah. just so you know the mechanism for example. Yes.
What was in the summer? I was like,
Like starting at 8:30. Well, there's a group exam. So we started. So you start at 8:30. You would get 50 plus 20 classes, 75. And then coming at 10. You have to do the group exam. Yeah, schedule. You'll be here by 10. The only other way to do it is to have you all outside of class.
Thank you.
This covers the reading assignment. I did the whole thing. Oh yeah, this is the first day. No, we didn't do any quizzes. The only thing we did was the seating chart. You could have a quiz anywhere. There's probably going to be a quiz everywhere. But I don't know if it's going to be. So they're not scheduled. It's just based upon the reading assignment for that day. Like for today, I think we're going to read chapter 4 or 5. So there's a synopsis recording on Monday. Education recording on Wednesday. We've only missed 2 lessons. So we look at the stuff on Moodle. And there's, so each unit has an exam sheet that goes with it. So that's the study guide for each exam. Study guide for the neurons and yeah, the syllabus, all the exams are scheduled. They're all on Monday. So you'll take an individual exam from 9 to 10, and then from 10 to 11 you'll take a group exam. So you'll be put together in that order at the same group exam. And that's where 10% will be.
On the last cycle, do we need to know the exact step of what's going on, or just
Well, you didn't know the exact steps up until the crossbridge. Crossbridge is the same,
the crossbridge is the same, but okay.
except for the last one.
Last part, we need to know.